Okay, we're going to go ahead and get started with our live stream. Good evening, everyone. On behalf of the South Texas College Biology Department, we would like to say thank you for joining us for our Spring 2024 Speaker Series. In a moment, we will introduce our speaker for the evening, but before we do, we'd like to let you know that this session is being recorded and will be available to you on our YouTube channel, so you can access this on a later day if needed. While our speaker is presenting, feel free to ask any questions about the topic, and our team will be in the comments. So we ask that if you could please leave your name, probably your first and last name, just in case your question does get selected. And for the title for today's presentation, it is as follows: Application of Molecular Diagnostics on Pests of Agricultural Importance. The name of our presenter today is Roxanne Ferris. She is a biological scientist for the United States Department of Agriculture, also known as the USDA, Animal and Plant Health Inspection Service, APHIS, Plant Protection and Quarantine, Science and Technology, Insect Management, and Molecular Diagnostics Laboratory, located here in the Rio Grande Valley in Edinburgh, Texas. Over the past 21 years, she has worked on the development and testing of molecular diagnostic methods for various types of arthropod species that are considered detrimental for U.S. agriculture. Her presentation, entitled Application of Molecular Diagnostics on Pests of Agricultural Importance, will provide an overview of the S&T agency and the molecular methods that have been developed to support diagnostics of pest species. She will discuss the highly important pest, the old world bollworm, and the development and validation of real-time PCR for its detection. Okay, Roxanne, whenever you're ready, you can go ahead and start.
Okay, thank you, Elizabeth, for that introduction. Again, as she said, my name is Roxanne Ferris. I am a biological scientist with the USDA. And just to simplify these long names that are listed here, I'm going to be referring to Science and Technology as S&T, and Insect Management and Molecular Diagnostics Laboratory as IMDEL. All right. So first of all, I'm very excited to be here. I was asked to do this presentation, and I jumped on the opportunity because it's very important for me to be able to reach out to students and the community to let them know what our lab here in the Rio Grande Valley does. There's always been a lot of curiosity when I mention to people that I'm a biological scientist. They always get excited and start asking me a lot of questions. Wow, what do you do? And where do you work? And so that's exactly what this presentation is going to be about. And so I'm excited to share that information with this audience. So thank you all for being here. I really do appreciate that. All right, so to start off my presentation, I'm going to give you all a little bit of background on S&T. All right, science and technology. So this is the mission statement here. Science and technology provides scientific and analytical support for PPQ regulatory decisions and program operations. S&T is responsible for ensuring that PPQ has the information, tools, and technology to make the most scientifically valid regulatory and policy decisions possible. S&T also supports PPQ, which is Plant Protection and Quarantine, program operations by providing practical tools for plant pest exclusion, detection, and management. So that, in a nutshell, is pretty much what our agency does. So we're basically the scientific backbone of the Animal and Plant Health Inspection Services. Okay, so one of the things that's really interesting is that our S&T lab down in Edinburgh, Texas is one of several laboratories located throughout the nation. So you can see on the map here that we have 7 different locations, and we actually have a sister laboratory to our lab located in Phoenix, Arizona. All right. So there is another S&T lab located in Fort Collins, Colorado, and that one is the Pest Identification Technology Lab. So you see each one of these labs has different emphasis on different topics. So we have one in Buzzards Bay, Massachusetts, which is the FPML, which is the Forest Pest Methods Lab. We have one in Maryland, which is the PPCDL, which stands for Plant Pathogen Confirmatory Diagnostic Lab. We have one in Miami, Florida. All right, so that one is the TIML, which stands for Treatment and Inspection Methods Lab. Now located in Raleigh, North Carolina is the office of our Deputy Administrator that heads all of our agency of S&T. And we also have the Plant Pest Risk Analysis Group in that location. Okay, so let me focus now on our lab specifically and our location. We are, as was mentioned, we are here in the Valley. We're located in Edinburgh, Texas. And if anyone is curious as to where we're located, we're actually located at Moore Air Base. So if any of you all are familiar with driving in the outskirts of Edinburgh heading towards McCook, we're out going that direction. We're about 14 miles northwest of Mission, Texas. So we're way out there in the country. And so it's interesting being in our location because it was once an active air base, an active military air base, I should say. And so when you're driving onto the grounds of the base, you can still see some of the abandoned buildings that were once used for the active military base. We still do have a runway that is an active runway. And so that is used mainly for planes that are releasing sterile flies out into the environment, which is a whole other section of work that is done here at this lab. So now I will get onto that. And as you can see the picture here on the right, we're a pretty decent size as far as the personnel. We're a mixture. We have scientists with different expertise. We have biological science technicians. We also have a large number of cooperators that are employed from UTRGV. We have some other universities represented as well, such as Baylor, and we also have North Carolina as well. So overall, as a lab, there are several areas of research or topics of emphasis, and those are highlighted here under the picture. And I'll go over those. So we have technologies to reduce the spread of citrus greening disease in plants. We have the pest detection and identification, which is molecular diagnostics, which is the group I'm in. We have pest management. We have biological control, and we also have sterile insect technique. All right. So now I'm going to focus in on the molecular diagnostic team, which I'm a part of. So we consist of 4 biological scientists and currently 4 biological science technicians. We will be adding an additional technician to our team soon. So within our group, we manage a good number of projects. So we have 9 internal projects, which basically means they're being managed by science and technology funding. And then we have 49 external projects, and these represent cooperative agreements that we have with other universities or with state agencies. So as a group, we work on development of molecular diagnostics for bacterial pathogens, and this is to maintain fruit fly colony health. We also develop various molecular diagnostic tools for arthropod species, such as fruit flies, beetles, and mites. We also work on development of CRISPR technology and nanoparticle technology for targeted treatment of citrus diseases. We also work on methods to improve sterile insect technique, or SIT. And we also work on identifications of captures and intercepts of important pest species and bacterial pathogens that could impact the fruit fly rearing facility. So a little bit about myself. I realize that the majority of my audience right now are high school students in their junior and senior year and some college students along with some faculty and staff. So one of the questions I do get quite often is, how did I become a scientist? Like, what did I do in my life and in my um, education to get to where I am today? So I'm going to talk a little bit about myself, and my background, and some of the traits that I feel I have gained throughout my years, so that helped me along the way. So to start off, back when I was a young girl, elementary school, I'll say, I was always interested in science. And I've actually always had a love for science in itself and also for science fiction. And I believe that's really where this all stemmed from. So, what used to be considered a possible phase by my parents actually continues on to this day, as far as my love for sci-fi and science and all that fun and good things. So, from an early age, I was watching Star Trek, loved Star Wars movies, anything sci-fi related, you name it. I was attracted to it, and so I always had always an infatuation with that, and I was always um, just amazed by some of the terminology and the methods that I would see them doing in these movies and shows. And yes, I know they were fiction, but still they had some reality to it. And I always envisioned myself that, hey, maybe one day I could do that. And so that love of science fiction, it turned into a love of science. As I got older into high school, back when I was in high school, there was not as much of an emphasis on STEM as there is today. I get that. And that's okay. I mean, it was still available to me. And so when I had options to take certain classes, because I had extra available time slots in my schedule, I always chose to take the sciences like physics, astronomy, advanced biology and chemistry classes. And so that in itself was obviously very helpful and helped me to become well-rounded in the sense of the sciences. And so one thing I would like to reiterate, and I do say this to a lot of students, is that don't downplay the arts one bit, because just as much as I was involved in science, I was also involved in the arts. And what I mean by that is that I was heavily involved in UIL competitions, not just the academic ones, but I was also involved in prose and poetry readings, impromptu speaking, duet acting, solo acting. And I will say this, that played an important role in my development as a person and also as a scientist. And you probably ask, well, how does it do that? Well, look what I'm doing today. I'm giving a public talk to this audience. And so part of those, I guess, experiences for me back when I was in high school was part of what helps me to be better at what I'm doing today, as far as public speaking and being able to share with you all the work that I do at a level that is where I'm able to communicate well to different levels. And so I always encourage students to try your best to get involved with those sorts of things, because it's going to help you develop your speaking, your ability to speak in front of audiences. It's just going to help you become more well-rounded. One thing I also do discuss, and this was outside of school, was I started playing piano at a very young age. So by the time I was in high school and into college, I was getting very advanced. And I always felt like that was a big part of my success in my sciences and mathematics. I loved mathematics, but I was never strong in mathematics. And I always attribute my strength, which came later, to mathematics and me getting more advanced in piano. And so there's a lot of studies out there that showcase that playing certain instruments, and piano being one of them, and certain other instruments, you are utilizing both sides of your brain. And so when you do that, it's exercising both sides of your brain at the same time, and that allows you to start looking at things and maybe understanding things that you maybe couldn't have understood prior. And I actually did see that with mathematics, because as I started getting more advanced, I started realizing that, hey, I'm able to understand some very advanced concepts here that I wasn't grasping quite easily before. And not only that, but it just continues to train your mind to always have that creative aspect to things. And so you're not always thinking straightforward, you're thinking outside the box now. And that in itself has allowed me to become a very successful scientist, because as I'm developing methods and coming up with projects and coming up with troubleshooting for certain projects, sometimes a lot of that we have to think outside the box to figure things out. Otherwise, you're sort of stuck. And so I always attribute that to my background. And so I always encourage people that yes, it's wonderful you're into science and that's great, but don't downplay all those other aspects as well, because they're equally as important in your development and hopefully in your future career endeavors. So some of my education, I graduated high school. I went on to get my Bachelor of Arts in Biology from UT Austin, as you can see in the picture there, doing the Longhorn sign. And that was an amazing experience. And so I fell in love with biology even more. Obviously, I was exposed to a plethora of courses out there, and it was just an amazing experience. And I wanted to continue pursuing biology. I really loved it, and I decided to work on my master's. And I came back to UTRGV, which was at the time University of Texas Pan American, but now UTRGV. And I was very fortunate to work in a laboratory with Dr. Dan Murray, who I know is listening today. And so I was very fortunate because he actually had a molecular lab that focused on bacterial genetics. So I learned so much in that environment. And my thesis, which was studying a global transcriptional regulator of E. coli, really, with that thesis, that type of work, I had to really get in there and learn so many new techniques and so many processes that I still utilize even today. And so that was a wonderful thing that happened for me. So with that, I've been here with this S&T agency for about 21 years. And I did start off as a biological science technician. I worked my way up in becoming a biological scientist about 7 years ago. I might be a little off on that. And since then, I've been leading various molecular diagnostic projects for the agency. And most of them are for developing routine diagnostics of intercepted specimens, or just diagnostics for vectors of pathogens, I'm sorry, vectors of diseases for plant pathogens. We have basically a good group here. We study various methods. We see how we can apply them for species identification, and we do validation of these methods. Sometimes we do tech transfer of these methods. Along with managing all of these in-house projects, I also manage a number of cooperative agreements, some with UTRGV. I have cooperative agreements with other universities outside of Texas, actually, with Cornell. I have University of Colorado. I have cooperative agreements with federal agencies as well. We have California Department of Food and Agriculture. We have I have agreements up at I mentioned Cornell and CDFA, and we also have some in Washington State. And so there's just a number of them. And so sometimes they involve hiring of technicians who come in as cooperators. Sometimes they're hired at their universities and they stay there. So it just sort of varies depending on the needs of the project. So what I'm going to do on my next slide is talk a little bit about, or show you really, some of the pests that I've worked on. And so these are a few of the pests that I've actually worked on while I've been here at this laboratory. And so there are a few that are fruit flies there at the top. And one of the main ones I've been working on recently, and I'm going to elaborate on that one a little further, is the old world bollworm. It's a moth you see there on the top right-hand side. I've also worked with mites, all right? And those, I mean, that picture of the mite is actually very enlarged. It's actually very tiny. You actually cannot really see it with the naked eye. You have to look under a microscope to see that one. And I've also worked with Swede midge. I've worked with chili thrips, also known as Sciurtothrips dorsalis. And I've also worked with the capra beetles. Okay. So I'm going to discuss a little bit of the molecular diagnostics that we do in our laboratory, just to give you all some background and some ideas. So when I mention certain things, you'll have an idea of what I'm talking about. And before I get into all that, I just want to emphasize the importance of why we use DNA. And this is a good example. So what I want you all to see is, okay, the picture on the left is an adult Mexican fruit fly. So I don't know if you all have ever seen one of these. You can see them sometimes here in the valley. And what we have on the right is it's the same species. It's the Mexican fruit fly, but that is in the immature form. Okay. And so what I'm showing in this example is that morphological identification, which is identification based on the outer appearance, so on the physical characteristics, it actually is possible for this fly, but you still have to have some expertise in order to be able to do that, because a lot of these flies, even though they're different species, they might look very similar, but only an expert can really see that, oh yes, I can pinpoint this one's different from this one. But for the immature phases of this fly, as you see on the right, it's very challenging to be able to tell species A from species B, because they look identical, I should say. But with the use of DNA, that changes everything here, because with DNA, it's magic, right? It's allowing us to be able to differentiate between these immatures where previously we couldn't. And so the nice thing about DNA is that we are able to do that with different technologies. All right. So these molecular tools, and when we say molecular, it's because we're relying on DNA, they're able to provide us with methods that are fast and they're accurate. So with DNA, like there's a saying that you've probably heard, is that DNA doesn't lie. And so that is pretty much true. It's going to be a straightforward answer, whether it is or it isn't a certain species. Okay. So one of the first things that we do in order to get to the point where we can study the DNA is what we call a DNA extraction. And as you can see from the picture, there is a pest. There's a cartoon image of a pest that is about to get extracted. Now, what happens actually is we take pests that have already been dead in alcohol, and we place them in these tubes and we crush them. All right. And we have certain instruments that we can use to crush them. The idea is that we want to expose the insides of the insect to the point to where the tissue breaks apart and maybe some of the cells are getting exposed. And that is not where we stop, actually. There are some more steps that we need to do to get to the point where we get to DNA. And we use what are called DNA extraction kits at our lab. And it's basically a kit where you're applying different chemicals and reagents to the squished insect and taking it through a series of centrifuging steps, which is spinning and collecting reagent, maybe spinning again. And there's some filters in some of these tubes that's collecting things that are important. And sometimes you're getting rid of things you don't want. So there's a combination of these sorts of steps. It's like you're following a recipe, like I said. And in the end, you're going to get some DNA. There's an image right there on the left. We have two technicians there that are performing some of these extractions on some pest species. All right, so once we do an extraction, the next step we would like to do is what's called a PCR. Now, I know many of you have heard the term PCR. It stands for polymerase chain reaction. And I know especially with this pandemic we had, you started hearing the term PCR probably a lot more often. because a lot of the diagnostics they were doing to determine whether or not you were positive for COVID were PCR diagnostics, all right? So that is a lot of what we do here at our laboratory. So once we extract the DNA from an insect pest, we apply that DNA to PCR, and PCR is basically a copy machine for DNA. And it's just going to make millions to billions of copies of a single strand of DNA. And I will mention it's not the entire genome of the insect, but it's a certain section of the genome. And so when we are setting up our PCRs, we hone in to certain sections of the genome by designing our PCRs to encompass certain small DNA molecules known as primers, and they act as targets to certain sections of the target DNA that we're wanting to amplify. And so once we do that reaction, well, let me go back a little bit. Well, we set up the reaction and we actually then place it into what's called a thermocycler, which is the machines you see there on the images and also on the right. And in the thermocycler, it basically takes the reactions through a series of temperature changes. And that is what starts up the cycling process for the PCR. Once it is complete, which usually takes about 3 hours or so, we should have a large number of copies of our DNA. And so the next step is, well, how do we know whether or not our PCR was successful? Because up until this point, everything's invisible. And so you're basically going by faith. And so when you take your reactions out of your thermocycler, they're going to look exactly as they looked before you put them in, which basically just looks like a little bit of water. And so not much has changed as far as you can tell. So what we need to do is visualize these PCR products by using agarose gel electrophoresis. And so this is a system where we load our PCR fragments onto an agarose gel. It pretty much looks like Jello, but imagine clear Jello with little tiny holes in the Jello so you can load your little product onto the gel. Now, there are certain things that we do to manipulate the PCR because obviously it's clear. We need to be able to see it. We add dyes. Another thing we do add is a chemical known as ethidium bromide, which does just so happen to be a carcinogen, but we do work on it quite often here in our laboratory. So the reason it's considered dangerous and a carcinogen is because ethidium bromide attaches to the DNA. It actually is, what it's called is actually it's intercalating into the DNA. So it binds into the DNA, but the reason we do that is because ethidium bromide glows under UV light. And so the idea is we want to have a way to visualize what we're seeing on the gel, and that is what we do. So we place all of these solutions together with our PCR product, we load it on the gel, we run the gel, so we allow separation of the PCR products because sometimes different PCR products are different sizes. And then we place it under UV light when the run is complete, and that will indicate whether or not we have successfully performed a PCR. If you see something fluorescing, you will have what's called a DNA band on the gel. And I'm going to go on to the next image so you can see that, what I'm talking about. So you can see right there, this is a gel showing some PCR products. Okay, so you can see the PCR products right here, and they're fluorescing. This gel image was taken under UV light. And so what I want to show you here is this is an example, this gel, of a chili thrips DNA marker that I designed several years back. And it's a conventional PCR-based system, very straightforward. So basically, if you get a PCR product for your DNA, that means it's positive for chili thrips. If you do not get a PCR product, which we see over here, there was something loaded on each one of these, okay? Just because there's nothing there doesn't mean it's just because it didn't work. We know it worked because these worked. We know here that those were not amplified, and that means that they are negative for chili thrips, okay? So that's sort of one way of analyzing PCR, a very straightforward way. So I imagine that possibly for COVID, it was done like something similar to this. However, I don't believe they were using conventional. They were using real time, and I'm going to get into that right now. So real-time PCR, it's another type of PCR. It's different from conventional, okay? So as I mentioned prior, when you're developing your PCR, there are several components that you put in your PCR reaction in order for it to amplify. So one of the components you put in there are what we call primers. And so those primers hone in to specific regions of your target DNA and they amplify those regions. So for real-time PCR, not only do you add primers, you add an additional component known as a probe. And a probe is just like a primer in the sense that it's a small sequence of DNA that hones into a certain region. But the difference is that probe has on it a fluorophore that will fluoresce upon DNA amplification. All right. So the advantage of using real-time PCR is that you can visually see when your DNA is being amplified as it's happening. So unlike conventional, where you sit and wait 3 hours and you have no idea what's happening, you have to wait the 3 hours and then load it on a gel to be able to visualize it and see whether or not it worked. With real-time PCR, that eliminates the need to have to, first of all, wait as long as the 3 hours, and it eliminates the need of having to perform a gel electrophoresis. So that, I guess, is where the name, you can say, comes from, because you're seeing the results in real time. And so the nice thing about this also, as I mentioned, it is very fast. So a typical real-time PCR can be about 45 minutes to an hour, depending on your assay itself. So that is obviously a huge advantage, along with the fact that you do not have to run agarose gels. And of course, it minimizes your risk of being exposed to that dangerous chemical, ethidium bromide. All right, so I am going to move on to another method that we do use in our laboratory, which is DNA sequencing. And so what you're seeing here is a chromatogram, and this is actual sequence data. I took this image off of something I had sequenced, probably bacterial species, I'm not sure. And what we're seeing here are some peaks. All right, so you're seeing some peaks, and each one of these peaks, they're different colors, and I'm honestly not sure if you all can see that on your end, but there's 4 different colors there. We have green, we have red, we have black, we have blue, and as you can see on the very top, we have some letters that are corresponding to the same color as the peaks. So these letters are representative of the nucleotides that make up our DNA. So I'm going back to biology 101 here, right? So we have, in general, we have 4 base pairs, right? We have the thymine, the guanosine, the adenine, the cytosine. And so those base pairs are what make up our DNA. And what makes DNA unique between species, between populations and such, is that there are various sequences of how these bases are lined up or organized. So, for example, you might see that species A, for example, has this sequence that we're looking at here, but maybe species B doesn't have this little section here of TTAA. Maybe it has GGCC. So it's getting down to those specific base pairs of, between certain species. And again, I'm just giving a very general example, but we utilize sequence information for developing the tools that we develop. We learn from it. We gather information from it. I have spent countless hours going through sequence data and editing sequence data, because this is a very clean example that I'm showing you. We don't always get this lucky. Sometimes the DNA is very messy. Sometimes the peaks don't look as clean. Sometimes we get double peaks. And so a lot of what we do is we analyze forward and reverse sequencing. And so if you go back to, again, Biology 101, remember, DNA is what? Single-stranded or double-stranded? It's double-stranded. And so because it's double-stranded, we can go that extra, mile, and we can sequence both the forward sequence and the reverse sequence, which is what we call them. They're double-stranded. One is the forward, one is the reverse, and we can sequence both of them. They're going two different directions, and then we can align them. And sometimes one of the sequences might have some really poor data. I'll just shake my fingers here so you see that, but the other sequence is beautiful. It's perfect. So a lot of times we rely on doing those forward and reverse sequences because we maybe get poor sequence data for one of the sequences, but we get great data on the other one. So that can give us the answer to what the actual correct base is. And so I have spent countless hours going through sequence data and just correcting base calls that maybe were incorrect. And that can happen for various reasons. Sometimes you just have poor quality DNA to begin with. Sometimes the reaction just didn't perform as well as it should have. And that's okay. That's part of science. It's not a bad thing. You just go back, you repeat things if you have to, and that's the way it is. And so we design a lot of our molecular assays that I've been talking about based off of sequence data. Now, someone might ask, well, why don't you just go to the sequence data to begin with if you're getting, the most accurate data there? I mean, that is the data. Why don't you just go to sequence data to do your species ID? Well, we do sometimes. When there's actually no molecular diagnostic tool out there, sometimes all we have to do is rely on the sequence data. But I will emphasize to you all that that is a very long and tedious task. It can be done, but it does take time. But if we can develop faster methods off of the sequence data, we can have molecular tools such as the real-time PCR, where we can just get our result in 45 minutes rather than to several days of going through sequence data. So that should answer that question there. All right, so let me discuss a little bit about a current project that I'm working on. Now that I've gone through all of that background information, I really want to focus in on a project that I'm really proud of. We've done a lot of good work on this project the past several years, and it's on the old world bollworm, which was that moth I showed you a little while ago. And I'm going to discuss some of the molecular diagnostics that our lab was part of in the developing and the validation of this method. And then I'm going to discuss a little bit about how we've worked on transferring this method out to the real world. So some exciting stuff going on here. So again, here is an image of the Helicoverpa moth, Helicoverpa armidra. And before I move on, let me just quickly ask how I'm doing on time.
I was just mentioning that to my coworkers. You have about 20 minutes, Roxanne.
Okay, perfect.
Mhm.
All right. So as I'm getting into this next phase of the discussion, like I said, I'm gonna hone in on this one pest here. This Helicoverpa armidra is the scientific name, the genus species, Helicoverpa armidra. I'm not gonna go on, around saying that name. The common name is the Old World Bollworm, or OWB is the acronym. It is actually one of the most destructive agricultural insect pests in the world. One thing I will mention, I think I forgot to mention earlier, that just because our laboratory is located in Edinburgh, Texas, that doesn't mean that we just focus on problems that are here in the valley. Because we are a federal lab, we actually work on problems at the federal level. Now, I'm not going to say that we don't focus on things that are local, because I mean, we do, but we are not limited to that. So this is one of those pests. Yes, it can be caught, I'll say, locally at our ports, but it can also be caught in Miami. It can be caught in Chicago. It can be caught throughout the world. So this is something that I just wanted to point out so you realize the impact of the work that our laboratory does. All right, so geography. it is located throughout the world. I won't read everything here, but you all can see it through Asia, Europe, all the way to Dominican Republic. It is actually not established in the United States. So I just want to make that clear. It has been caught, but it is not established, all right? So it has a very large host range. Over 180 host plants have been documented, which include corn, cotton, tomato, all the way up to cabbage and citrus. There's quite a bit. There's an image here I'm showing you. That is a corn, and you can see what looks like the little worm in there. That's actually an immature of the Helicoverpa genus right there. All right, next slide. So there are identification challenges to this pest, which is why we have worked on it. So as I mentioned, the really bad player, the old world bollworm, it's not established in the US, but guess what? There is a species of Helicoverpa known as Helicoverpa zea, or the corn earworm, which is actually established in the United States. Now, it's bad, but it's not near as bad as the old world bollworm, all right? So the problem that happens is that these two species, zea and armidra, right, the corn earworm and the old world bollworm, they look identical. So if you're looking at two adult moths, you will not be able to tell them apart, all right? So the same is actually true, and even more so for the immatures. Now, some people will actually go as far as saying that the immatures actually just cannot be identified morphologically. So that's where DNA comes in. We have to rely on the DNA for instances such as this. This is a classic example where we're not able to look at it and tell what it is. Is it the really bad player? Is it the one that's already here? We don't know. So we have to rely on DNA. And so that's where we rely then on molecular methods to be able to distinguish between these two species. And guess what we have? We have a real-time PCR probe assay. So this is similar to the real-time PCR I mentioned earlier. And basically, I'm going to explain here what we're seeing here. There's two graphs, and there's only one specimen that's being amplified in this PCR that I'm showing you here. So the one on the left shows an amplification growth curve for Helicoverpa armidra, the bad player, okay? So the green, and I don't know if you all can tell, you can see a green amplification curve, okay? This red one here, I'm sorry. Yes, I'm sorry. Let me go back a little bit. This red one here is the threshold, okay? But not to confuse you with the red amplification curve, which is our internal control. And then we have another color, blue, which is representative of our Helicoverpa zea, and that remained a flat line. Okay, so what we're seeing here is that Helicoverpa zea is not being amplified. It's remaining a flat line. And what we're seeing is that the Helicoverpa armidra is being amplified along with the internal control. Now, the internal control, we have to use them in every PCR reaction that we do. And so that will tell us if there's amplification of the internal control, that will tell us that the PCR assay was a success, all right? If there were no amplification of the internal control, then that would tell me, that would be indicative that somewhere along the lines, this real-time PCR assay failed. And even though I'm seeing a positive amplification for old world bollworm, I'm not going to take it seriously. It's not going to be confirmed until I repeat the assay and make sure my internal control works. All right, so on that same note, on this side, we're seeing an amplification growth curve, but this time for zea, which is the corn earworm. So you can see here the zea in the blue is being amplified along with that internal control, which is good. And we're seeing this time that the armidra is remaining flat. So that tells me this is a positive for Zea, and this one was a positive for our Midra. All right, so this is actually what we call a multiplex assay. And all that means is just a fancy word for saying that we are able to detect multiple species in one PCR reaction rather than having to do multiple. And so we have incorporated this assay where we have two different primer sets, two different probes, and we can throw everything in there. And if it's an Armidra, it'll detect it. If it's a Zea, it'll detect it. It's like an all-in-one. It saves us time. It saves us resources. So it's a wonderful assay. This assay actually has been validated at our lab by testing for accuracy, repeatability, and for sensitivity. I know I have to hurry here a little bit, but I want to talk to you all a little bit about why we do validate and the importance of validating these assays. So especially assays such as these, which might be applied in other laboratories or in the outside world, we have to validate. And one of the reasons is we need to make sure that the tool that we've developed is fit for use as a diagnostic tool for the said species, in this case, our Midra. And is this method successful at other laboratories? Because that in itself is extremely important. You know, if you develop an assay that only works in your lab, there's no point if it's not going to work in other labs. So we really need to make sure that it does. And if it doesn't, we work with that. We see what we can do to get things to work, to get things to be where they need to be. All right, so a little bit about the validation that was done when this paper was published. We were part of this publication as well. We did use a sample size of 452 specimens. 18 of those were species, variant species, and 28 different locations where these species were found. And so we did confirm the accuracy of this method by morphologically IDing, by dissection, all adults. So as you can imagine, that was a very tedious task, but they were all confirmed with morphological ID before we moved on to doing molecular ID. So we do that whenever we're developing for the first time a brand new tool for a certain species. So that basically gives us the justification, if you would, that what we're developing, the molecular tool, is based on sound science, that it was a morphologically ID'd specimen, so you know there won't be any inaccuracies. And that is important because if you think about it, if you're not going to be confirming by morphological ID, when you develop a molecular ID, how are you going to know that your molecular ID is correct if you never confirmed it with first morphological? So that's the idea here. So all the adults and larvae that were used in this study were sequenced for what we call the barcode region. So not to get too much into it, but the barcode region, it's a CO1, which stands for cytochrome oxidase 1 region of mitochondrial DNA. And it's a standard that's used worldwide. It's worldwide accepted that that's a certain region that is PCR amplified, that has been used across many species throughout the world. And there is a universal barcode database where when you barcode items, you can look at that bold, they call it, database, which is the barcode of life database, and you can determine whether or not your species matches with any of the sequences that are in that database. And that's a universally accepted database that has data in there that has been vetted, and we can confirm our diagnoses through that system. We also look at sensitivity, which is basically how far can we dilute samples to where they are no longer amplified with this assay. And so for this one, we were able to get down really low to 0.01 nanogram per microliter. And reproducibility, you always want to make sure that you can repeat this assay at different laboratories. All right. So this was also validated at our laboratory, but when we validated, we actually doubled the sample size. And so we had 13 different species from 24 different localities. The only difference was that at our laboratory, we didn't use adult specimens because we didn't have any to go with. So we utilized what they provided to us, the other laboratory, which used the adults. And then we took additional samples. We had larvae, and we sequenced them for the barcode region. And we compared those IDs to the bold database for molecular ID as well. And the nice thing was that we had no false positives or negatives that were reported using our validated assay. And also for sensitivity, we saw it was the same. It went down to the 0.01 nanogram per microliter. And for repeatability, I performed these assays in triplicate, and I saw that all across the board for all the armidras, we got 100% repeatability of the results for the Helicoverpa armidra. Overall, the test success was over a 98.6% rate of success, which was a really good success rate. So we were really happy with that. And so we were able to finally validate this assay. So what was our conclusion from doing this validation study? So we did these 2 validation studies that included 1,300 specimens. And so the results from both studies showed us that the assay is successful on different life stages, so both adults and immatures. It's successful on very small amounts of DNA, down to 0.01 nanogram per microliter. It is highly accurate at diagnosing a Helicoverpa armidra, or the old world bollworm, and we could get fast results that were generated within a day. And so when I say within a day, it's because we still have to include that extraction step and then the real-time PCR step. So once you include all of that work that's involved, we can have results within a day, as opposed to 2 or 3 days if you're using other methods. So this validated method has actually been applied at our laboratory. It's used for processing Helicoverpa intercepted specimens. So we routinely receive Helicoverpa immatures that are coming in from different ports of entry. Like I know just this past month, we received over 40 specimens that came in from the port of Miami. So we do routinely get these. And to date, I will say that we've processed over 2,000 of these intercepted specimens. Now, I will make a note that all of the specimens that have been identified as Helicoverpa armidra have been coming from areas where it is known to occur. So far, we're still not seeing any from our neighboring countries or from the US. All right. So I do have some images here of 2 of our biological science technicians, Rosie and Tony, who have been heavily involved in the performance of the DNA extractions for these Helicoverpa immatures and for performing the real-time PCR assays at our laboratory. Okay, I'm getting towards the end here. So one of the biggest success stories that we have here at our lab is that we've been able to transfer this technology successfully over to a plant inspection station. So what we have shown through this project is that with proper training and proper instrumentation, we set up an actual molecular lab there at that plant inspection station at Los Indios, Texas. You can see that this method, it can be transferred outside of our laboratory. And so, and in FY24, actually just a few weeks ago, this method was officially transferred over to the Los Indios PIS. So what is the benefit of doing this? Well, the benefit is that the molecular ID can actually be performed right at the spot of, or closer to where the interceptions are occurring. And so that is going to be a benefit in the long run. It's going to save on the time for the time of waiting for the turnaround time for results, for the shipments that may be placed on hold when there's an unknown specimen that they're trying to wait on an ID for. And so there's a lot of advantages to this. And so we're really excited because this is the first of, we're hoping, many technologies that can be transferred to PIS areas. And so with that, I think that is my last slide. And I just like to thank everyone for being a part of this presentation today. And if you have any questions, I'm happy to answer any. And if I can't, I can always try and figure out what the answers are and then get back to you all. I have my email there in case anybody ever wants to reach out to me with any questions or further information. I'd be more than happy to answer your questions and so forth. So with that, I am done.
Okay. All right, Roxanne, really quickly, I'd like to compliment you on your speaking skills. You're such a great presenter.
Oh, thank
And
you.
we can kind of tell throughout the comments, like when like students are really engaged, and depending on their questions,
Yeah.
we can tell, I guess the level of engagement we can see within the comments. And so for your talk, a
That's
lot
awesome.
of the questions were answered out throughout your talk. And so I'm going to mention one
Okay,
and then I'm
good.
going to add a little bit to it. So this question is from Michael Gonzalez. He says, do you collaborate with local ag businesses and farmers for your studies and research? And so I'm going to add a little bit to that because I know you mentioned this insect, although it might not be hurting us directly, right, in our area, because you do get samples from all over the place.
That's
I'm
right.
gonna ask this, how are growers, and not so much locally, but just in general, the ones that are getting affected, how are growers adapting to this insect since it has, or sorry, since it has like a large host range? Because I know locally
Yes.
in the RGV, there are some practices where they will literally put like a net over the citrus trees to prevent them from getting, infected with something. So what practices have you seen that have been implemented, like to try to get this insect away from their crops?
Right. That's a good question. Unfortunately, like I was saying, the Helichrysopus, which is the corn earworm, it's already established here in the United States, but it's not nearly as destructive as the old world bollworm. So that one is still not established in the United States. There have been a few detections, and when they are, they quickly quarantine the areas and they do what's called surveying, and they do survey the areas to see if they can, well, what they do is they set up traps for these moths and they see if they can detect any more that are in the surrounding areas. And I know not our lab, but there's other labs that have been involved with processing all the survey material that has come in, which has been apparently in the hundreds to thousands of processing these to determine if they detect a positive find. But in the meantime, I will state that a lot of growers, and not just for this pest, but like let's just say for others in general, there are techniques that they use to monitor their fields. I know that they use traps. I know that there are certain, I guess, agents, pesticide agents that are, I guess, approved to be used to control certain pest species. In some cases, and I didn't get into it in this talk, there are forms of biological control that are also used for a number of pests and diseases. And so there's also SIT, which is a sterile insect technique. So for example, I will talk, I guess, a little bit about the Mexican fruit fly, which is Anastrepha ludens. It was in a picture here in one of my slides. That one is found here in the Rio Grande Valley. We try to control it, the population, because it's established in Mexico. It does come across, and we try to control it by releasing sterile insects. We actually here at Moore Air Base, as it's not part of S&T, but it falls under plant protection and quarantine. We have a rearing facility right next door, and what they do is they rear sterile Mexican fruit flies, and they sterilize the males, all right, by irradiating the males. And remember, I mentioned that we have an air base here. Well, we still have an active runway, and we have planes that take these massive amounts of sterile flies and they release them into the environment. And so what that does is when the sterile flies from the rearing facility mate with the wild females, which are females that are naturally found in the environment, they do not produce offspring. And so that is a way of controlling the population levels. And so they release these flies very often. And so that is happening right next door. So there's a lot of different methods that come into play as far as for controlling the situations that whatever, depending on what area farmers or growers are in, depending on what pests are big in their area, there's going to be maybe not just one method, but maybe they use several combined methods to control the situation.
Okay, thank you. And
Sure.
I am glad that you did mention like a little bit elaborating about citrus, because the citrus industry down here is
Yes,
huge, right? Billions of dollars. It's a
absolutely.
huge industry. So thank you for mentioning
Absolutely.
that.
Yeah, and that's
Okay.
one of the things, like when we mention the importance of these agricultural pests, they're important because they're damaging very large amounts of commodities. And so whether, let's say it's citrus, I mean, we're talking in the millions, they can lose money. And so that is a big impact. And we obviously don't want to ever lose, for example, our ability to drink orange juice. We take it as it's a luxury right now, right? We don't want to lose that luxury, and we're hoping that doesn't ever happen. But we have been impacted by citrus diseases, and it's something that I, as of right now, with what I do, I'm not directly involved in the disease aspect of things, but a lot of my colleagues are, and so it's something that we're definitely overall all involved with.
Okay, and then I'm going to combine our last question. We
Sure.
have two students. One is Valeria Rodriguez, and the other one is Liliana Gonzalez. So the first one is like short, right? Like why insects? What inspired you to work with insects, right? Out of everything. That's one part. And then the other part,
Okay.
what was your experience as a woman in the STEM field? How did your gender impact your career?
Okay.
So you got
So
it,
let me
insects and then your experience as a woman in STEM.
okay, let me start off with the insect one. And I'm just going to be quite honest here. I actually did not choose to work on insects. And it sounds kind of funny to say that. They sort of chose me. I actually had my training in a bacterial genetics lab or molecular biology lab. And so one thing I love to emphasize to students is the fact that once you learn the techniques of DNA and all the methods of how to play with DNA and to analyze DNA, DNA is universal. And so once you learn how to manipulate DNA and study DNA and analyze DNA, you can apply that to any field. And so I was trained on applying it to bacteria. And there was an opportunity for me here. They had some technician positions open, and they were in a molecular lab. Well, I quickly learned that the only difference between working with the DNA from bacteria and the DNA from insects is the way you retrieve the DNA. And so the method of extraction is going to differ. So same with plants. The method of extraction is going to differ. And I've worked with plants as well. So once you obtain the DNA, that's where all the fun begins. And everything you learned, whether you learned it on bacteria, or if you learned it on insects, if you learned it on plants, if you learned it on fungi, what have you, you can apply it the same way. It's universal. So insects chose me, and I fell in love with it. I just through my years working here, I've gained a lot of knowledge. I've worked on a number of different insect pest species, and you start to learn the impact that certain pests have on certain fruits, on certain vegetables, on certain areas. And you meet researchers from around the world. When I worked on thrips, I loved working on thrips. I had an opportunity to go into an international meeting. I met people from all over the world. I collaborated with them, and I learned about all the different species of thrips they were working on and the problems they had in their country, whether it be in the Netherlands or in China, Japan. I was getting insects from all over. We all collaborated together to develop diagnostic tools, and I really loved it. It was so much fun. And that's how it is. You develop, these cooperative relationships because everybody is focused on the same problem, whether it be here or somewhere else. We all come together and we're working together on that. And so that's how it happened for me with insects. And so as far as the other question, it was how did being a female affect my career in STEM? Was that the question? I wasn't sure.
Yes, let's see. What was your experience as a woman in the STEM field? How did it
Okay.
impact your career?
I will say that it has been challenging. I'm not going to lie. Um, I am up until 3 weeks ago, I was the only female scientist here at our laboratory. We've just recently got another female scientist. So that, is something that is, I'm thinking, going to change very soon. I have noticed throughout my career from the beginning to now, from when I first started going to scientific meetings, I'd be one of the few females. And not only that, but I was one of the few Hispanic females because I am Hispanic. And yeah, so it it is challenging, but I am a very driven person, and I never saw it as a wall. I basically saw it as an opportunity to open the doors for, I would hope, for future female scientists, and to hopefully pave the way and lead the way and to guide other young females into joining this field. I would hope that by seeing me in this position, it can serve as a sort of as a source of, as what's the word I'm looking for? Guidance in a sense like that. Oh, or, someone, I guess, to look up to in a sense that, oh, well, if she can do it, I can do it too. And I'm from here and I'm local. I left. I did leave for a few years, but I came back. And at the beginning, I didn't think there was opportunity here, but a lot of that has changed. Our lab has grown tremendously, and the university is growing tremendously here. STC is growing tremendously, I'm seeing. And so I feel that now a lot has changed, and there's a lot more opportunity here for local scientists to be a part of this exciting field. And so with that is how my career has kind of gone. I'm not saying that's how it has been for everybody, but like I said, I have been here for over 20 years. So I've seen a lot of changes in my time. And so I think it's all been for the positive. And so I'm excited about that. And I look forward to a new generation joining us. Definitely. I hope that answered the question.
Yes, it did. And we're so happy to hear that. And the words like
Yeah.
encouraging to see
Yes.
a woman in STEM local,
Right.
you're empowering young women as well. So
Yes.
congratulations. And we're so
Thank
happy
you.
for all your success. I'm gonna
Thank
close
you.
out our presentation. So we hope that this presentation was enlightening for you all, like such as maybe you weren't aware of this type of research that we have happening in our backyard here in the Rio Grande Valley, or also how like genetics can be intertwined with agriculture. A lot of people will kind of breeze over agriculture and not think much of it, but there could actually be a lot into it, such as molecular genetics, and maybe you weren't aware of that. So we hope that this talk kind of opened your eyes. And if you are interested in learning more, she does have her email here on the screen. And we can also see, maybe we can also have a cooperative with the USDA one day. We'll see. That would be
I think
nice.
that would be something to look forward to. I would actually really like that. I think we can look into doing something like that. And I will just reiterate, just if you all are interested, get my email address. And if you all have any other questions, you can always feel free to email me, okay? I'll be here to answer your questions.
And I think that's it, guys. We hope you enjoyed tonight's presentation. Look out for future talks from us in our speaker series, and we hope that you all